This recording is the 6th part in recordings covering the lymphatic system, but I'm actually going to break it up into part 6A and part 6B. 6A is going to cover cell-mediated immunity, while 6B is going to cover antibody-mediated immunity. Now, the cell-mediated immunity is accomplished by the use of T lymphocytes, or T cells. And we've discussed the specific types of T cells, but let's discuss them again. So the first type is your cytotoxic T cells, and I'm just going to abbreviate it like this. Those are the ones that are going to attack cells that are foreign to your body. It's recognized that this doesn't belong here, or cells that are your body cells that, though, have been infected by a virus. We will talk about those cytotoxic T cells in this recording. Another type of T cells that we'll talk about in this recording are the helper T cells. Now, those helper T cells help to activate B and T cells, so it'll be talked about again when we talk about antibody-mediated immunity, but it also activates your T cells. Now, the suppressor T cells, I'm not really going to discuss today, so they inhibit the function of B and T cells. The memory T cells, I'll just say in passing. So you'll see I'll mention the memory T cells. So they respond to a previously encountered antigen. Now, the T lymphocytes, they won't attack, or I should say, even though they like to respond directly by cell-to-cell contact to potential threats, they do require either an antigen-presenting cell or the presence of these MHC proteins. And so I gotta mention what these MHC proteins are. So MHC proteins are short for major histocompatibility complex. Sometimes they refer to them as human leukocyte antigens. They code for self-proteins. They determine you. And a T cell learns to recognize you, me, as MHC, and they will ignore it. But if an MHC protein is got an antigen associated with it, the T cells will recognize it as now being abnormal and will attack if it recognizes something that doesn't belong there. So I want to talk about different types of MHC proteins and how they're associated with antigens. So the first type is a class 1 MHC protein. Now, these proteins are found in all of your nucleated cells in your body. They will pick up little fragments of antigens if the cell, say, becomes infected, and put it up onto that surface of the class 1 MHC. Now, if that antigen's abnormal, the cell's infected, that will trigger activation of T cells. But if the peptide that's associated with that class 1 MHC protein is normal, the T cell will ignore it. Say, okay, I'm just going to bypass you. Now, class 2 MHC proteins we find on antigen-presenting cells and lymphocytes. Now, if, say, for example, you see here an antigen-presenting cell, which was a phagocyte, is engulfed a pathogen that you see here, it will destroy it using the lysosomes, and some of the fragments from it are going to come up to the cell surface and associate with that class 2 MHC molecule. And that's going to be, this cell is going to be an antigen-presenting cell. And what's going to happen is it will present the antigen to a particular type of lymphocyte. And so lymphocytes can interact with class 2 MHC molecules. And we're going to look at kind of very shortly how certain T cells know to interact with certain cells. And it all comes down to these other markers that we're going to talk about. In order to recognize the antigen, you have these CD markers, or CD stands for cluster of differentiation markers. The two that I want you to remember are the CD8 and the CD4 markers. So cytotoxic T cells have CD8 markers. And these particular cells, because they have a CD8 marker, they're allowed to interact to anything bound to a class 1 MHC protein. So remember, class 1 MHC proteins are found on all nucleated cells of your body. So if there's an antigen present on that class 1 MHC protein, a cytotoxic T cell can come around, check it out. And if it recognizes that particular antigen, if it's specific for that particular subpopulation of T cells, it will respond and kill that cell. Now, the helper T cells, which are a very important T cell population, because it's like that merging or the kind of bridge between the cell-mediated and antibody-mediated immunity, plus it really is going to be very important in your innate immunity, and you'll see very shortly, is the helper T cells have these CD4 markers. Well, they can interact with anything that is associated with the class 2 MHC proteins. So remember, class 2 MHC proteins are found on your antigen-presenting cells. So those are actually going to be presenting the antigen to the helper T cell and say, helper T cell, you need to get some of your buddies to help. And his buddies would be like your cytotoxic T cells or maybe your B cells. And so the helper T cells will be interacting with anything with the class 2 MHC protein. So what we're going to look at first the activation of a cytotoxic T cell. So with this scenario, you have to present the antigen. In this case, what's presenting the antigen is in an infected cell. It is not like a macrophage. It is a cell that has become infected. And so what's happened is we're going to zoom in on the infected cell here. And so here it is the infected cell. And right there is the MHC class 1 protein. And what do you notice is bound to it is a particular antigen. So all these are the antigens. So what it's doing is it's flagging these cytotoxic T cells, come over here, check me out. If you recognize you need to kill me because I am infected, I really don't want all my other friends, nearby neighbors to get infected. And so what we have is a cytotoxic T cell right here. And remember, we have multiple subpopulations of cytotoxic T cells. They're going to be specific. Some populations are specific to, say, mumps, some for measles. So this cytotoxic T cells coming up and say, oh, I can talk to you because you have a class 1 MHC protein. And the reason why it can get close and personal to it is because of this marker. That there is your CD8 marker. So anything with a CD8 marker, which we find in cytotoxic T cells, can interact with things with a class 1 MHC protein. So here it comes up, and right there is a T cell receptor. And that T cell receptor is going to recognize only certain specific antigens. In this case, this particular T cell receptor recognized that antigen right there and said, I know you, I know what to do. And so what's going to result is we're going to have activation of that T cell. And as a result of the activation, you're going to have increased cell division. And so divide. Some of them are going to stay behind as our memory cytotoxic T cell in case later on and they're going to be inactive. They're kind of sleeping. They're like Z's, okay? So they're sleeping in case you're exposed to the antigen again. But the rest of them, you got tons of these new cytotoxic T cells that are, again, specific for this particular antigen right there. And what we're going to do is these are going to go around, find all these infected T cells, and they're going to destroy them. And they're going to destroy them, try to get it multiple ways. They're going to release perforins, which disrupt that cell membrane, triggers that lysis of the cell. Release these factors called cytokines. It's kind of saying, hey, you might as well give it up. You need to die off. And so it triggers what we call apoptosis, which actually is cell suicide. It also releases toxins. They call them lymphotoxins because they're coming from lymphocytes. And that disrupts the metabolism of the cell, which also all this contributes to the destruction of that infected cell. Now, what about the helper T cells? What are their role? Well, helper T cells are going to be active or activated when a antigen presenting cell presents it with an antigen. And so here in this case it's a dendritic cell, which is an antigen presenting cell. it's phagocytic. It eats this bacteria, presents some of the antigen to the cell surface. This helper T cell says, ooh, I recognize you. I'm going to go off and get more help, because they're going to recruit a lot of help from a number of different areas. So let's look at the scenario where we're going to recognize the antigen by a helper T cell, or that CD4 positive T cell. So in this case, the red dots are antigen, and I'm just going to abbreviate antigen as this, okay? And this here is an antigen presenting cell. So the antigen presenting cell, and I'm just going to put APC for antigen presenting cell. We're going to zoom in, and you notice right here, the little red dot is the antigen. This here is the class 2 MHC protein that has the antigen associated with it. It's going around and saying, hey, I need to find some helper T cells to help with this. We got some stuff going on. And so here, this, I'm going to circle it. Actually, I'm going to erase this so you can see. So right here, that is your CD4 marker. So right in here, this is your helper T cell. So that helper T cell with that CD4 marker knows I can go and talk to anything with the class 2 MHC. So I'm going up and say, hey, what's up? And you see this antigen, it's like, no, I recognize that particular antigen. I'm going to get ultra excited, and I'm going to become activated, and I'm going to divide, and I'm going to go and get some help. And so this activated helper T cell, you'll divide. Some of them will be inactive and become memory helper T cells, so they're sleeping. So I'll put these little Z's again. But the active helper T cells are going to do a number of different things. They secrete cytokines. So when they secrete cytokines stimulate both cell-mediated and antibody-mediated immunity, both. They promote division of T cells and maturation of cytotoxic T cells. They're going to bring in some of the innate immune response. So it attracts phagocytes to the area and stimulate them to work. Say, hey, keep going, we need your help. They stimulate and activate natural killer cells, which again are part of your innate immunity. They promote activation of B cells, which would be your antibody-mediated immunity. And subsequently, those B cells will start producing antibodies. And so the second part, we're going to be looking at the antibody-mediated immunity and tying in, again, these helper T cells, because those helper T cells are needed to kind of get those B cells to start to become activated. So this is the end of the first part of part 6. So this is part 6A. So we're going to do in part 6B, look at antibody-mediated immunity.